Good afternoon, everyone. And the first thing I would like to say is a big thank you to David and Andy for inviting me to speak. I've had a very interesting time so far. It might go downhill from now on for me because I actually have to do some work and do some talking. I'm going to talk today about some work that my group has done in the area of antibodies, and in particular, the relationship between some of the very new data that's coming, and then we have huge amounts of data on, so being able to do IG-seq, being able to get all these antibody sequences, and how you might be able to use that data if you think about it in a structural sense, instead of just thinking about it in a sequence sense. So I'm sure most of you are familiar with this, but antibody next-generation sequencing has really changed the amount of data we have available and probably should be changing the way we are doing antibody drug discovery more than it already has. It's relatively straightforward to get very large snapshots of of antibody sequences in somebody's repertoire. So you can get snapshots that can be tens of millions of sequences. And you can do this to sequence, say, the naive human antibody repertoire. So what do people have inside their repertoire? Or you can look at pre- and post-immunization data sets, so you can start to see what has been raised by a vaccine, so what comes out. Or you can do the sequence repertoires from different species, because if you want to understand what a human antibody is, it's probably helpful to understand what a not human antibody is as well. One of the things that I think is important to remember about this data is, of course, it's always still a snapshot. The theoretical antibody repertoire that each one of us is carrying is much, much larger than any of these numbers. So it sounds like we have a massive set of data. We do, but it's still only a sample of the real data we want to deal with. And the second thing to remember is that most of this data still is unpaired. So if we're talking about standard human antibody sequences, we're talking about we've sequenced H chains, so the VH, and we sequence the VL, but we don't know which VH goes with which VL. Now that's not an insurmountable problem, but it does mean this data isn't sort of perfect in any sense yet. So what we did this year, in fact, we released it earlier on this year, was try and find out how much of this data was available. So we created a database called the Observed Antibody Space. It's basically just a huge collection of all the publicly available IG-seq outputs. And here we focused on where people have sequenced the entire region. So not just if they sequenced just, say, the H3 region. We want where they've done the work and sequenced the entire VH or most of it, so we can use that. We discovered that IG-seq outputs are currently fragmented across lots and lots of different repositories. They tend to be presented as raw nucleotide reads and are actually quite difficult to process in any kind of way. So we've now collected 57 studies into here. So those studies might have lots and lots of samples within them, but each study, so some of the studies have got tens of millions of sequences within. That's more than 500 million antibody sequences that are just there for people to look at. And they cover diverse immune states, organisms, and individuals. About half that data is human, to give you a kind of scaling, and a lot of the rest is mouse. And then there are other species that are in there. The idea was we have cleaned and sorted this data, so it's annotated, translated, and numbered. So you know that the data is all totally consistent, and you can say, I want all of the VH sequences, and I want to see them all lined up and numbered by IMGT, and I can see what residues occur at a given IMGT position, for example. And it's all completely freely available, so you can download the whole lot if you want, or you can explore it in different ways as you might find interesting. Now, in some sense, this was quite a big divergence for my lab, because my lab is primarily interested in structure. And when we think about antibodies, we think about them from a structural viewpoint. So we tend to think in terms of this diagram over here, the three-dimensional structure of the antibody, because I would say it's three-dimensional structure that governs how it's going to behave in the system. So it's three-dimensional structure that says whether it will bind to something. It's three-dimensional structure that gives it its biophysical properties. And you can infer some of those from just knowing the sequence, but not everything. So what we really wanted to be able to do was combine these two things. Can I put that structural information onto these very large-scale data sets? And if I do that kind of thing, how much does it help me? What can I learn from doing it? So to give you an idea of why we think this is important, this is just a very simple example. In that picture in A, I have two VH chains. They've both been crystallized, okay? And if you look at the H3, which is purple in one and blue in the other, it's basically the same shape. What is interesting about this is no sequence method on Earth would have told you that it was going to be the same shape, because they've got different V genes, different J genes, so you're not going to pick it out like that. The sequence identity between them is less than 30%. Actually, if you go and look even closer at this, you can see that the same types of residues, so if you like the same chemical pharmacophores are pointing out from this loop in both these cases as well. So actually, if you were looking for the same antibody, that's it, but you couldn't find that if you didn't know the structure. On the other side is the opposite end of the spectrum, which is where you have two antibodies that by most measures people would go, these are going to be very, very similar. So there are only two changes between those H3 sequences, and yet they are structurally completely different. So it is highly unlikely that they're going to behave in the same way when you think about binding. Now, we've used these kinds of ideas to look at post-vaccination data sets, so particularly post-vaccination from flu, and you can start to pick out very interesting patterns of the usage of H3 shape that you can't see from H3 sequence. But that's not what I'm going to talk about more today. So what I want to do now is I want to link my sequence information to that structural information. And we start with that with something we call our structural antibody database. So SABDAB, we built this about 5 years ago now, and we built it for ourselves, really. So anyone in this room who does work with protein structure has probably been to the PDB and has probably done something which I did very naively. You type the word antibody into the PDB in the naive belief that it will give you a list of antibody structures. The reality is, it doesn't. So what we built is a fully automated method that's able to check the PDB every week, strip out all the structures that contain antibodies, take those antibodies and number them, so collate them, curate them, and present the structural data consistently, mark out where the antigen is, mark out what is the VH chain, the VL chain. We also use this to pick up nanobodies, and we have a similar database that does this for TCRs. And you can see that this is actually quite an important effort if you're going to keep working with antibody structures, because I think it's about approximately 2% of all entries in the PDB contain an antibody. So there's a vast amount of structural data that is continuously growing. So if you don't stay up to date with it, if you're using a database that's a year out of date, you're probably certainly a few hundred antibody structures behind the curve. And you can see that a little bit from here. So that picture was a snapshot I took of the front page of the database back in July 2017. And you can see it doesn't even reach 3,000 structures there. I went and checked yesterday, so the last update then was on the 4th of October, and now we have 3,272 antibody structures. So this growth in data is just, it's ongoing and is going to keep happening, and we should have all that data if we want to exploit it efficiently and well. So my group has spent a lot of time building a lot of computational tools for doing the examination of antibodies. So things for like IPatch and EpiPred for looking at the paratopes and epitopes of antibodies and predicting them and trying to improve them. AbAngle for looking at the angles between the two domains and how you predict those. Anarchy, which is a numbering tool, and then a set of tools like Sphinx and Scallop and Freed for modeling the CDRs. What I want to talk to you about very briefly today is our modeling software for antibodies, which we call a bodybuilder. And the thing I want to sort of note right back at the beginning of this is there's nothing rocket science in this. So I would say each of the individual components here, they're good, but there are many other really good ways of doing this. So we built a tool which is a template-based method for modeling antibodies. We use our database, obviously, as the back end for that, because that's got the largest collection of publicly available antibodies. We can optimize the angles for the VH and VL, we can do our loop modeling, and then we can refine this and try and make as good antibody structure as possible. And then we've tested it like everyone else has. So we've done it on various benchmarks, but just to give you an idea of performance. So unfortunately, a bodybuilder was too new to be part of the antibody modeling assessment. That had already happened when we did this. So we did post-diction on that. We did only use structures available pre the contest, but I want to be quite clear, I wouldn't use this slide to claim that we were way better than Schrödinger or Rosetta antibody. I want to use this slide to say we're about as good as everyone else out there. I happen to think we're a little bit better, but I wouldn't put lots of money on that particular fact. So looking at this, these are the targets for the AMA2, and these are the RMSDs for the whole thing, and then various regions, the framework, and then the CDRs across here. And if it's green, you're doing pretty well, and if it's sort of red or yellow, you're doing less well. So you can see we're doing about as well, or maybe a bit better than everyone else. So the question is now, why have I bothered telling you about a tool which I've said, yeah, it's quite nice, and yeah, it works as well as everyone else's? Well, it's for two reasons. The reason we built this tool was to get these two facilities within it. One is speed. The average runtime for model generation is just over 30 seconds. That's good. We can actually do it faster than that now. Rosetta antibody is tens of hours at their best estimates. A recent paper suggested you needed 200 hours per model. What that means is that if you had an IG-seq data set, you can't model it with Rosetta antibody. Nobody's got a big enough computer. You can model a few thousand with a pretty big computer. What we can do is model it. The second part of this, which was a key step we wanted to put in, is we estimate the accuracy of modeling. Now, if you're only interested in an individual antibody, that's not that interesting because you just want a model of that antibody, and if it comes out as being a poor model, well, there's not a lot you can do about it because you still want a model of that antibody and you're going to use it as best you can, but you're just going to go, well, it's a bit bad, but I wish it was better. If you're modeling hundreds of things or thousands or millions of things, this means you can choose to not model the stuff you will model badly. In other words, don't use that. In much the same way you would say, if I can't run this experiment, I'm not going to do it. So here we're saying, I can not only model very fast, I can choose to only model those things I believe I will get good results for. And that combination allows you to think about modeling very large data sets in very clever ways. So one of the ways we've used this is to try and build a human antibody model library. Sounds kind of a bit silly, but if you think about it, if I wanted to describe the sequence space of antibodies now, you would go potentially to our observed antibody space database, and you'd be able to look and see what residues are used at every single IMGT position for 250 million antibody sequences. So you have some idea of what sequence space for antibodies looks like. That doesn't tell you what structural space for antibodies looks like, what the variation in properties are. So we took a particular data set. We've done this with a couple now, but this is the first one we did. It was IG-seq data from about 50 healthy individuals, and it gave us, we had about 13.5 million unique sequences. And what we did was we reduced the volume of those sequences to about 20,000 structurally diverse antibodies that we knew we should build good models of. So I'll give you a little more detail on how that works. So what you start with is unpaired data, about 5 million heavy sequences, about 10 million light sequences. The first step we do, because it turns out it's the fastest step we can do, is we check how well we can model the CDRs. And we just keep those where we think we can model the CDRs accurately. Because if we can't model them accurately, then actually, even if we build that model, it's useless in our library because it's not going to be informative about the structural space. And that takes our numbers down quite a lot. The next thing we do is we cluster those sequences. And we're really clustering the frameworks there. And we're trying to remove things that are very similar to one another. Now, all of these things are going to have effects and add biases to our data. And then we end up with a much smaller set of heavy and light chains. We then test all the possible pairs of those, and that's 24 trillion calculations. It gets a bit boring, but computers are quite fast. And you can identify those which are reasonably sensible pairings, and there are plenty of very good ways of doing that. And then we estimate what we think the structures of those CDRs are, which is what we had at the top. So I still haven't built a model. And I do a structural comparison, and I pick out about 70,000, and I actually build basically 70,000 models, and then I structurally superimpose all of those and take the 20,000, it actually ends up about 19,000, because I use a particular cutoff threshold, that are structurally diverse. So these are structurally distinct from one another. So what I'm hoping there is I have a representation of the variation of structure that you can see across antibody space. Now, there are clearly things that I haven't captured. So, for example, the length of H3 here is somewhat limited because we tend to model very badly for long H3s. So this set will tend to have shorter H3s than you would expect in a general human data set. Now, we've used that data set in many different ways, but perhaps the way we've used it most recently is something we call the Therapeutic Antibody Profiler. And this is an idea of doing something like Lipinski's Rule of 5. So the concept of Lipinski's Rule of 5 was, I'll look at things that are drugs, and I'll see what makes them safe as kind of oral drugs. Not by doing any chemistry on them, just these things are all drugs, and they all have less than 5 hydrogen bonds exposed, for example. Yeah? Or I have particular values of log P, they're all lower than a particular value. And the question is, could I computationally do that with antibodies? Now, obviously, these values are not going to be, if you like, as straightforward to calculate, but you can do a similar kind of thing. And our analog here for the drugs is a set of clinical stage therapeutics. So that's what we're going to use. We're going to calculate metrics on those, and we're going to say, I expect things that would also be other good clinical stage therapeutics to fall within the region of these ones. So not to have properties that are really different from anything I've observed before. So our data set. Well, the first data set we used was 137 post-phase 1 clinical stage antibody therapeutics that actually come from a PNAS paper in 2017. We built models of all of them, and everything I'm doing I'm going to use models for, and there's a good reason for that, because if I'm talking about screening very, very early on, you're not going to have a crystal structure of that candidate. You're going to be able to build models of that, but not going to be able to build crystal structures. So everything I do has to be accurate on models, not on crystal structures. Because if I already have a crystal structure, I've already probably done lots of experimental testing and other things. This is designed to be much earlier in the stages, and that's something you can check. And the models are accurate enough for our metrics, so we tested it with the 56 of these which have known structure. And for context, on the back of this, we also put the distributions of the models of our human antibody model library. And that's partly to answer this question is, and I think the answer is relatively obvious, but it was clear that many people hadn't thought about it before, would any old human antibody make a good antibody therapeutic? And the answer to that question is no. But it'd be useful to know why the answer to that question is no, because they're clearly safe because they're inside your body. But that doesn't mean that they are a good therapeutic for other reasons. So if I start with the easiest and simplest metric to explain. So this is CDR-H3 length, and in fact we actually use total CDR length. So that's the length of the 6 CDRs all summed together. And the first thing you see is that CDR-H3s of clinical stage therapeutics tend to be shorter than you have in general human IG-seq data. And I don't think that's a massive surprise to anyone. It could be for a number of reasons. It could be reducing entropy loss, because obviously if you have a shorter set of loops, that's that. Or it could be something that's helping to prevent aggregation. But I don't need to know why. I just need to know that's the distribution you tend to see. So if you have H3s that are much longer than this, you're probably going to struggle to be a therapeutic. We actually chose to use the complete length of all of the CDRs. The second metric is something that people also tend to associate with the quality of antibody therapeutics for various things, which is patches of surface hydrophobicity. So if I get you to look at the picture that's on the right-hand side of the screen, what I have here is the antibody chain, and what we have here is it's colored, the residues are being colored by their hydrophobicity. And you can see I have this big patch where lots of hydrophobic residues are together. And the idea of this measure is to measure exactly that. Not how much hydrophobicity do I have on the surface, okay? What's more problematic is how much hydrophobicity do I have clumped together on the surface. So having 5 hydrophobic residues on the surface is not a problem. Having 5 right next to one another is. And that's what this measures. So if I show you this as a distribution instead, so in blue, this is our patch hydrophobicity for the CDR vicinity. So it's the CDRs plus the residues around them and anything else which touches them. So you get an idea of the patch expansion across it. And reds are the values for the human antibody model library. So you can see that clinical stage therapeutics are quite clearly, deliberately engineering this down. Yep. So if you had a candidate that sat somewhere out here, you'd probably be a bit worried that it might not have some very good biophysical properties. The next set of metrics, the next 3, all relate to charge. So the first 2 of these are kind of the reverse of the hydrophobicity. So one for looking at patches of positive charge and one looking for patches of negative charge. So once again, if you have large clumps of charge on the external of your molecule, you can have complete effects there. And then finally, there's the structural FV charge symmetry parameter. So this is based on a measure that was actually invented in Ken Dill's lab, where what they did was they looked at the charge on the sequences of the VH and the VL and said, actually, there should be some kind of symmetry in this. So if one's very negatively charged, the other one should be very positively charged. And if you have too much disbalance, this will be bad. We've tried to take that one stage further with, rather than just thinking about the sequences, think about the structures, because it's only those residues that are surface exposed that matter in terms of this. So what we end up with is 5 metrics. You could have picked other things. I'm not claiming that this is the best possible list, but I can tell you that in this list you have things that kind of relate to the way we would think would make antibodies have good biophysical properties. So if we think about something like total CDR length, longer lengths probably relate to having aggregation or lower affinity or some form of convex binding site. Patches of surface hydrophobicity, if you've got too high a value, you might be thinking about aggregation or high viscosity. And your positive charge or negative charge, effects on expression and stability. And then there's sort of this colloidal instability and high viscosity that people have linked to structural, this charge symmetry between the two parts of the FV. But in no sense do I think any of these directly correlate with an experimental test for these. That's not what they're for. They're, in some sense, a hand-wavy way of saying, if I had 1,000 things that are starting to look interesting, I could very quickly say that 30 of those are probably never worth looking at again, or maybe 900 of them, because this is incredibly rapid. You can do it at any stage you want. There's nothing to stop you doing that. And the way we decided to use it was very simple, which is we would amber flag anything which was in the top sort of 5% of highest or lowest values we've ever seen, and we would red flag anything which was just above any value that's ever been seen before. And of course, it's up to a user to decide if you're red flagging and it's, you know, one length longer, so it's the total CDR length is 61, should I panic? Or should I be like, I can probably get away with that? Whereas if total CDR length is 80, you'd probably go, I'm probably not going to progress right now. And the same is true for the other metrics. So does it work? Well, first of all, validation, things it shouldn't flag. So what we did was we went and collected another 105 post-phase 1 therapeutics. So we just went and collected them from the literature. And we checked what happened. Only 8 of those got any flags. So 8 of them got red flags, which implies you won't pick out genuine therapeutics as having issues very often. What it also says, interestingly, is almost all the time when it was red flagging them, they were just on the edge of the distribution. So we weren't finding things that were a very long way off that had made it to be therapeutics. They were right on the edge. So the key one here, I think, is Irinoumab, its total CDR length actually is 60 according to our definition. And of course, it got flagged because our limit is 59. And that's, you know, correct. But it is also worth thinking about the implications of that. So now what we did was, of course, you can improve this method every time there are more therapeutics that come into the system. You just build models of them and you add it to your distribution. So now I can have 242 clinical stage therapeutics to make up my distribution. And I can go the other way. Things that it should flag. Now, it's slightly harder to get enough data here, but I've now got my distribution here, and I'm going to talk about just 2 case studies. So this is about a case study on an anti-NGF antibody. And the original antibody was this MEDI578, which was found to be minor aggregator, which I liked as language, and it sits here in our patches score for the hydrophobicity. They then made some mutations to it, which were to improve binding affinity, and they got MEDI1912. That's an extreme aggregator. Can I say that we have no surprise at all in seeing that? That's a very long way off our distribution. And then some larger number of changes to MEDI191 here, which doesn't aggregate at all, and that comes out with a green flag here. Or here, I can talk about the anti-IL-3 candidate. So the original was this, I think actually the original is this one, AB001, which had very, very poor expression. And then they made some changes to it and got, I think, this one, AB08, which had very good expression, but actually didn't bind as well. And so then they made further changes and ended up with this, which still had good expression. So it's not conclusive evidence that everything is going to work perfectly, but it does point to the fact that if things are sitting outside these distributions, you can tell that they're likely to have bad characteristics. And the key here is the speed at which you can do this. So you can do this at any point you like to be able to do these things. So finally, I guess my advert. This is the kind of output you get from TAP. We have a website for it. People can enter sequences. You can also speak to us to have it freely available for companies. We offer that out as well. There's a paper on it which is currently on the bioRxiv, but you can see what you get for a given antibody is a summary of the results, pictures that relate to this. This is actually a model that you can rotate and translate to look at whatever you want, and the flags marked on and where your antibody lies on all of those things. So I should finish by saying thank you to all the people who did the work and all the people who pay to allow me to do this work. So first of all, I should say a big thank you to all the companies and research councils that fund me, because otherwise I wouldn't get to do what is my favorite thing. And then in the 2 different projects, we had collaborations from MedImmune, GSK, Roche, and UCB in terms of TAP, and OAS was a collaboration with UCB. And then I should particularly mention Conrad, Alex and Matt, whose head you can just about see there, who actually did all of this work. So thank you very much.